Hello, my name is Enoch Yu. I'm a genetic engineer at the Appleby Research Institute. Today, I've been challenged to explain gene therapy in 4 levels of increasing complexity. Gene therapy is an emerging technique that modifies genes in cells, like replacing a broken gene with a healthy copy or introducing a new gene. This technique has a great deal of potential for treating diseases like cancer or hereditary conditions like cystic fibrosis. From this video, I hope everybody learned something about gene therapy at some level. Hello, Jeffrey. Are you excited with what we're going to be talking about today? I certainly am. We're talking about biology, right? Yes, have you heard about gene therapy before? I think it's something that can help and treat people, like physiotherapy, but I have no clue about how it works. Sure, let me explain. Have you tried cooking or watched your parents cook before? Yes, all the time. I really enjoy the tomato seafood pasta that my dad always makes. It's very tasty. Amazing. Usually for cooking, there's a recipe that contains a set of instructions on how to prepare the food. Your dad might have followed the instructions of a recipe to make your favorite pasta. But imagine that your dad uses a recipe with many problems in it. The recipe might fry the pasta, have spicy chili, or leave out something you really like, such as seafood. If your dad follows the recipe, it might lead to a dish that isn't so great. Fortunately, we can simply fix the recipe by replacing the wrong steps with correct ones, or adding in new steps that you want. So the pasta will become tasty again? Definitely. Now imagine that the recipe contains instructions not for preparing food, but for how our bodies work. Our body is made up of trillions of units called cells, and inside each, there's a recipe called the genome that instructs how they work. But sometimes there are errors in the recipe, or something useful is missing from it. Scientists could use a technique called gene therapy to fix the cell's recipe by replacing the wrong parts or adding in new parts. This is just like how we could fix the pasta's recipe. That sounds cool. Once the cell's recipe is fixed, wouldn't they become healthy and stronger? Yes, mistakes in a recipe could cause the cell to function wrongly and lead to many diseases or sicknesses. By fixing these mistakes, the cell's functions could be restored, and these diseases could then be treated. Also, adding new steps to the recipe could boost the cell's functions, so they can fight diseases better. Can gene therapy treat anemia? My father told me that he has sickle cell anemia, but that I don't have it. Certainly, sickle cell anemia is caused by a part in the cell's recipe being wrong, but gene therapy can replace it with a correct part and therefore treating sickle cell anemia. Such treatment has already been done on babies since 2018, and has the potential to completely cure sickle cell anemia very soon. Isn't that amazing? It sure is, but what if you accidentally fixed the recipe wrongly? Sometimes I try changing my dad's recipe for it to be better, like adding in ice cream, but it can taste really bad. That is a very smart question. Many things can go wrong with fixing the recipe, like changing the wrong steps or accidentally introducing a bad step. But scientists can fix the recipe many times and test them all to make sure that after being fixed, the recipe works well. It's just like how you could tweak a recipe many times to find the one that works best. By a lot of testing, we can make sure that gene therapy is safe and functions properly. But wouldn't that require lots of time and effort? It does, which is why gene therapy is quite difficult and costly to develop. This form of treatment is still in its starting stages and not widely used. Hopefully, in the future, gene therapy could be developed to become more accessible and help improve the lives of patients all around the world. Nice to meet you, Albert. Have you ever played a video game before, such as Minecraft? Definitely, it's a very fun game. Did you know that behind every game, there's a complex program that provides the instructions for how the game behaves and works? In the program, these instructions are made from a language called code. So the process of mining blocks in Minecraft is controlled by code? Exactly, but imagine that there are problems with the game on your computer. A feature might be broken with glitches, or there might be a missing part of the program that prevents you from opening the game. You might also feel that the game has a missing feature that another game has. How could you solve this? I'm not sure. Can we correct these mistakes by editing the code? Yes, programs are written in code, so editing the code could fix the program. Say you have a working version of the game on another computer. You could get a small piece of code that works from that game and replace it with the piece of broken code on your computer. To add another feature from another game, you could just add the code from that game onto the code on your computer. That's amazing. This mechanism looks very useful. And it's also how gene therapy works. In the cells of our bodies, there are instructions that make us who we are and determine how we look, function, or behave. These are found in the genome of all cells. Have you learned about the genome and what it is? I sure have. Aren't they a very long sequence of nucleotides that determine traits like hair or eye color and also control the operations happening in the body? I think that it is needed for creating proteins. That's correct. Our genome is made out of double-stranded DNA located in the nucleus. Genes contain information in the form of nucleotides that encodes for an amino acid chain. This information is read by proteins called ribosomes, which creates that amino acid chain, which then folds into a protein. Isn't that the central dogma of molecular biology? It's how DNA is used as a template for mRNA during transcription, and that mRNA codes for a specific chain of amino acids that is translated by ribosomes. Ribosomes read mRNA in codons of 3 nucleotides, and the combination of these nucleotides determine the amino acid added onto the chain, or if the chain should start or stop. You sure learned a lot from biology, but the gene might mutate and change the sequence of nucleotides due to things like UV light, radiation, or an error when replicating the DNA. These mutations could be passed down from parents. All of this might lead to defective or missing genes being present since a person is born or developed when the person grows. Do you know how this would affect the protein made from the gene? I think mutations might alter the amino acid chain made. From biochemistry, I learned that the function of proteins largely relies on their structure, which is formed from amino acids interacting with each other, like hydrogen or covalent bonds, such that the amino acid chain folds together and obtains a 3D structure. Mutations could change the amino acid chain, which would change these interactions and lead to a different protein structure. Is that right? Yes, mutations could lead to a change in the amino acid chain, like altering specific amino acids, making it end too early if it is a nonsense mutation, or completely changing the amino acid chain if it is a frame shift mutation. This change could result in a different protein structure that makes the protein fail to function. If the mutation occurs at a place in the gene corresponding to where the amino acid chain starts, the chain might not even be made at all. In short, though mutations might change nothing, like silent mutations, they might also lead to non-functioning and possibly harmful proteins or the protein not being made. This could result in devastating consequences, like cancer. Have you heard of it? Yes, isn't it when cells keep growing without stop? I've also read that a mutation in the P53 gene is a common cause of it. True, the P53 gene codes for a protein that regulates how often a cell replicates, but if there's a mutation in the P53 gene, it will result in this protein failing to function or being missing. So the cell would grow uncontrollably and lead to cancer. Couldn't scientists, like fixing an error in a game, get a healthy P53 gene and use it to replace the mutated P53 gene in cancer cells? Also, can we add new genes into the genome of cancer cells that code for cancer-suppressing proteins? Spot on, that is gene therapy, or modifying genes in the cells of our body to treat or stop diseases. Just like fixing a game by editing the code, gene therapy can fix cells by editing genes. This would restore functioning proteins or introduce new beneficial proteins in the cell, which can treat a wide range of diseases, like cystic fibrosis, a fatal condition that damages the lungs. It is caused by mutations that result in misfunctioning or absent CFTR proteins. But gene therapy could insert a healthy gene that replaces the mutation and can restore these proteins, therefore treating this disease. Is that something you are researching about? Correct. In my lab, my team has been working on developing a system that replaces the broken gene for CFTR proteins with a healthy one in human cells. I've heard that cystic fibrosis is a hereditary disease, which should be because genes are passed down to offspring, and the mutation could then be passed down with them. Does that mean gene therapy could also treat other hereditary diseases, like sickle cell anemia or muscular dystrophy? Certainly. Also, if gene therapy is performed on a person at the earliest stages of their life, like in the embryo, it could remove the disease-causing genes in all of their cells and stop the disease from being passed down onto the cell's offspring. So, the patient and their children would not have the disease. Wow, so gene therapy can basically eliminate a hereditary disease? That's really amazing. Let's start off with a question. Do you know what is gene therapy? Is it editing the genes in cells, like replacing misfunctioning genes with healthy ones? The root cause of many problems or diseases, like cancer or sickle cell anemia, is due to defective genes, which leads to faulty or missing proteins. This causes the disease. I think that because gene therapy directly targets the root cause of these diseases by restoring proper genes and therefore functioning proteins, it makes the treatment very effective. That's correct. This aspect of gene therapy allows it to tackle diseases that were very difficult or even impossible to treat before. Also, gene therapy could introduce new genes coding for helpful proteins that treat diseases or enhance the functions of cells. For example, these genes could cause the cell to produce cancer-suppressing proteins or make immune cells better target pathogens. Now that we talked about the goal of gene therapy, do you understand how the desired gene is delivered to cells? I think that genes cannot just be injected into cells, but need to be carried into the nucleus of the cell by something called vectors. One common vector I have read about are viruses, like adenoviruses or retroviruses, as they have evolved to become very skillful in inserting their genome into the nucleus, so the virus can hijack the cell and replicate. This virus could be injected intravenously into the target tissue, bloodstream, or lymphatic vessels. Exactly. It is also very important to modify the virus beforehand so it doesn't cause harm. In my lab, the virus's genome can be genetically edited to contain a desired gene with CRISPR. This desired gene could then integrate with the cell's genome by a process called homologous recombination. Do you know what this is? It is when two very similar DNA sequences exchange strands, right? This could happen when the desired gene is sandwiched between two gene sections from the cell's genome. So the overall gene is very similar to a part of the cell's genome. From homologous recombination, both switch with each other, which leads the desired gene on the cell's genome. By editing the two gene sections that sandwich the desired gene, scientists can adjust where this switch occurs. This is necessary for locating the broken gene to replace it with a healthy one, or adding a new gene at the right spot where it is most useful. Right, some viruses like adeno-associated viruses promote this process. Integrating the desired gene into the genome is very helpful, as the gene will be replicated along with DNA and the cell's offspring. This mechanism is crucial for curing hereditary diseases, because if gene therapy was performed on embryonic stem cells, all the cells in the baby from those cells will contain the gene. So the disease is treated, and the baby's children would also not inherit the disease. But wouldn't editing the germline raise many ethical problems? It can be justified to use gene therapy on babies for severe or incurable genetic diseases like cystic fibrosis or muscular dystrophy. What if people use gene therapy on other undesirable genetic traits, like if a person gains weight easily, finds it hard to build muscle, or has poor skin? These traits could be argued by some as genetic disorders, but by others as cosmetic enhancements and are unacceptable for gene therapy. Also, these changes from gene therapy have the potential to change one's life negatively, but the individual produced from the germline has no choice to consent or refuse treatment. For example, there is no guarantee that gene therapy works or is safe all of the time. There is a risk that the gene is inserted wrongly and disrupts other genes, possibly promoting other diseases like cancer or leukemia that can ruin the individual's life. It is certainly very difficult to draw a line separating what gene therapy should and should not treat. While most genetic traits like eye color are barely understood by science, let alone change them, it is only a matter of time before we do. Also, though gene therapy has only been tested on somatic cells and not the germline so far, it is very possible for it to happen soon. These ethical considerations make it certainly necessary for strict regulations to be put in place, like guidelines to follow or an ethics board that approves any gene therapy treatment or research. All therapies, especially those for the germline, must undergo plenty of testing and fulfill many requirements before being used. I really agree with the mechanisms that you suggest. All forms of gene therapy should have the ultimate goal of improving the health of people, and not for materialistic aspects like becoming more beautiful. But what are your opinions on the cost of gene therapy? As you know, current forms of gene therapy require hundreds of thousands to millions of dollars. Would gene therapy only be accessible to the wealthy and not the poor? Sadly, yes, it is an unfortunate reality that not everyone can have gene therapy, which will lead to wealthier people living longer and healthier lives than the poor. While there are organizations that could sponsor treatments for those who need it, this might apply to only a select few of people. Hopefully, innovations in the future could drastically reduce the cost of gene therapy and make it available for all of us. Good afternoon, Greg. I am sure you are very familiar with gene therapy and used it in your research. That's right, my lab focuses on developing gene therapy treatments that tackle cancer, specifically the genes that regulate glycolysis. Glycolysis is a metabolic pathway that converts glucose into pyruvate and is a crucial part of cellular respiration. Decades ago, it was observed that cancer cells had a peculiar way of metabolizing energy. Even with plenty of oxygen, cancer cells more readily use glycolysis rather than oxidative phosphorylation to obtain ATP, despite how glycolysis is far more inefficient. Isn't that the Warburg effect? Recent research has found that many cancer cells share mutations located in the genes that regulate glycolysis. Such mutations are thought to impair the control of glycolysis, so it occurs abnormally often, while also causing many cancer-related functions like preventing cell death, or promoting metastasis, or the spread of cells around the body. Exactly. Our team aims to replace these mutated genes for glycolysis with healthy versions of those genes, which would make cancer cells return to normal and function properly. We plan to genetically edit a retrovirus vector so it contains the healthy gene, but also displays proteins that bind with cancer cells. So the retrovirus can attach onto cancer cells and deliver the healthy gene that would eventually integrate with the cell's genome. Gene therapy will be performed by injecting a batch of these viruses into the tissue around the tumor. That sounds amazing. Using virus vectors for gene therapy is really fascinating, as the viruses can be genetically engineered to bind with and deliver genes to only a particular type of cell, like cancer cells or lung tissue. This will not only make gene therapy more effective, as it has specific targeting, but also prevent harm, since gene therapy could be damaging to cells that it is not intended for. Virus vectors also allow for gene therapy to be performed on cells in areas very hard to access, like internal organs, with minimal invasiveness and no need to perform complex surgeries. The virus vectors can travel to those areas through the blood or possibly through lymphatic vessels. Yet, there's still many aspects of gene therapy that need improving. Viruses have a limited gene carrying capacity of around 4,000 bases, which mean they cannot carry more useful gene sequences that are too long. This problem greatly limits the capabilities of gene therapy and is an obstacle from it treating more diseases effectively. For example, large-scale DNA sequencing found the OCTL3 or organic cation transporter-like 3 gene, which is around 12,000 bases long. This gene can effectively trigger apoptosis, or cell death, specifically for cancer cells. I thought of introducing this gene into cancer cells by gene therapy, but with current techniques I could not because the gene does not fit into the virus. Also, while the disease-causing genes in viruses can be edited out, it is very difficult to edit out all the antigens or proteins on the surface of the virus. Our immune systems can identify these foreign antigens and spark a strong immune response, causing inflammation, fever, and more. In severe cases, fatal problems like organ failure could occur as a result. For example, in 1999, a patient testing gene therapy for a liver disease died from the unwanted immune response caused by the vector. Another problem is if the virus was used before, the immune system could recognize the virus and kill it before it delivers the genes. This would prevent repeating the treatment many times, which is a major problem because most forms of gene therapy require multiple doses. Certainly, this is an issue that cannot be ignored, but there are solutions. Patients could take immunosuppressive drugs during treatment, or researchers could inject the smallest dose of virus that is effective. Researchers could also opt for virus vectors that are less likely to trigger the immune system, like an adeno-associated virus. Sadly, there is no way of completely eliminating the risk of this immune response. Also, it is almost impossible to understand all the complexities of the human genome, and editing it with gene therapy could result in unforeseen consequences. For example, what if the new gene disrupts the function of other genes? What if the new gene codes for a protein that binds with and deactivates another protein? This protein could have a very important function, like regulate cell division or respiration, and deactivating this protein could cause serious conditions like cancer or organ damage. As such, gene therapy could make the situation even worse. That is why testing and experimentation is always crucial. Gene therapy treatment should also be tailored for and tested on the patient's genome, because these unforeseen consequences are different for everyone. Adding a new gene in the same location might be harmless for one person, but cause immediate cell death for another. Therefore, gene therapy typically does not allow for an one-size-fits-all approach. Each treatment must be conducted individually and case by case, for every patient to ensure its safety and effectiveness. This is a key reason why gene therapy is so expensive and takes so much time and resources to research and develop. But despite these problems, we should always stay optimistic. The potential for gene therapy is, without a doubt, life-saving and limitless. With advances in gene editing and delivery happening frequently, it is just a matter of time before this form of treatment is used to improve the health of billions of people around the world. Gene therapy is definitely something that's going to change the world, and I am really glad to be working on something so significant like this. This is gene therapy explained in 4 levels, and I hope you learned something interesting today. Thank you for listening.